Hello, my name is Cody Matching, and this semester I did the effect of alcohol exposure on the developing zebrafish HPA axis. Specifically, I looked at larval zebrafish, and I worked alongside Dr. Lamora. So for my introduction, basically the HPA axis, or the hypothalamic pituitary adrenal axis, is responsible for the secretion of numerous hormones, most notably cortisol, and it regulates um, functions such as sleep, temperature, behavior, and these are all controlled by the central nervous system, or the CNS. But one of the most studied functions of the HPA axis is the stress response, which utilizes the hormone cortisol. During the stress response, the hypothalamus releases a corticotropin releasing factor, which cues the anterior pituitary gland to secrete the adrenal corticotropic hormone, and this stimulates the adrenal cortex to release cortisol. Fetal alcohol spectrum disorder, or FASD, is an extremely dangerous and damaging disorder. In fact, I've seen some studies that's shown that 25 to 50% of women in certain areas drink while pregnant, so that's very concerning. And these can actually cause behavioral disorders, anxiety disorders, and learning disorders. And extensive research has been conducted over the years to better understand the specific effects on alcohol and how alcohol has on the developing fetal brain. And depending on the length of exposure, for instance, how long the mother drinks and how much the mother drinks during pregnancy, the effects of FASD can be mild to like just social disorders or some slight panic disorders to severe, which is actually inhibiting intelligence and learning. Zebrafish have actually been used as an ideal model organism for FASD research is because they reproduce really quickly, they have high numbers of offspring because they lay hundreds of eggs, and they're just really cheap and cost effective to maintain, and they actually have very easily traced neural pathways, and we can observe them and we know a lot about their genome, so they're the ideal organism for this study. And research conducted by Baggio et al. In 2018 has shown that the treatment with alcohol on zebrafish embryos around 24 to 26 hours post-fertilization, or HPF, at concentrations from 0.1% to or 0.01% to 1% volume per volume concentration of an alcohol solution at 95% ethanol for 2 hours cause a significant increase in anxiety-like behaviors for the zebrafish larva. So the objectives of my research was to basically, when I completed this, I wanted to allow for the examination of the impact of alcohol on different stages of fetal development. Very specifically, I wanted to see what is the critical time point of development. So what hour of development does a critical thing happen, which alcohol exposure would be absolutely devastating to the organism. And another impact of this study I'd wanted to get from this was a better understanding of the underlying causes of behavior observed, and it could be used for children who are afflicted with fetal alcohol syndrome. So our hypothesis was that the zebrafish treated with alcohol will have a lower cortisol level than the control fish due to the damage to the HPA neural pathways. So these fish will be less anxious, and this could explain deviance in kids potentially with fetal alcohol syndrome. And the three aims of this study are to examine the behavioral effects of the fish treated at discrete time points, to observe the cell death of the HPA neurons as per acridine orange, and to examine the cortisol levels in adult fish with stress. Unfortunately, due to COVID-19 outbreak, we were not able to complete the acridine orange staining and the cortisol level analysis because we didn't have access to the labs because they all closed. So our methods: in order to determine the exact time interval in which the HPA pathways develop, 4 groups of 50 zebrafish larvae at 22 hours post-fertilization, 24 hours HPF, and 26 HPF were tested for the thigmotaxis and phototaxis responses, which are 2 key behavioral assays that have been well studied. The larvae were exposed to alcohol concentrations for 2 hours at a concentration of 1% volume per volume. And after the 2-hour interval passed, the alcohol-treated larvae were moved back into the normal non-alcohol-treated tank and then moved into another tank as to help ensure the fish got into pure tank water and no alcohol or as low alcohol as possible. And the control were also had this done to them, but with no alcohol in order to ensure the placebo was done and no extenuating variables. In order to test the phototaxis, 50 zebrafish were placed in their own 4,000 milliliter fish tank, and a 1,000 lumen LED light was shined on the fish tank, which was half lit up and half darkened. After 1 hour, the number of zebrafish who left the area of light were counted, and the higher percentage of fish who left the area of light indicated the higher levels of anxiety. This is due to the fact that when zebrafish feel stress, they move away from the light in order to alleviate that stress. In order to test the thigmotaxis test, each group of zebrafish was placed into their own tank. The larvae were directed toward the middle, and then the table of the bowl was hit 2 times with a fist in order to simulate the thigmotaxis response. So basically, they're directed to the middle where I swirled them to the middle, and then I hit the table 2 times, and then that vibration of my fist and the sound caused them to, if they properly exhibited the response, they would go toward the edge of their tank, and that's called thigmotaxis. And after the hits, the number of fish were counted after 6 seconds, so we gave them 6 seconds to react, which is, a reasonable amount of time based on current research. And the larger percentage of fish that moved to the edge indicated higher levels of anxiety. So if the fish lingered in the middle, they weren't as anxious, therefore they didn't feel the need to move, which would indicate an inhibited cortisol response. So the results, this is my figure 1. They all were significant. Each value was significant, but the most significant we found was the 26 hours post-fertilization. The blue bar represents the control groups. The orange bar represents the treated groups, the alcohol-treated groups, and the y-axis is the percentage that exhibited the behavior of thigmotaxis. So, for instance, the 22 hours post-fertilization control group exhibited nearly 90% of the fish exhibited the thigmotaxis behavior, in contrast with the experimental group experiencing around 45%. But you can see that 26 hours post-fertilization was the most significantly impacted by the alcohol treatment, with only around 12 to 13% of them exhibiting it, as opposed to control being around 90%. And for the phototaxis results, no significant results was seen. They all exhibited low amounts of phototaxis, which was very interesting because they're in contrast to research conducted by Bai et al. In 2016. He actually found that larvae zebrafish should exhibit a positive phototaxis, but when I shined the light on the LED light on the tank and I gave them an hour, none of them moved into it really. They all just kind of stayed where they were at. They actually avoided the light, it seemed like, most of them. So no significance was found with that result, with that test. So alcohol exposure at 26 hours post-fertilization induced the greatest developmental defects on zebrafish based on the thigmotaxis behavior. So this could be indicative of something going on at 26 hours post-fertilization that is causing, like a critical time point in development, that is causing the greatest effect on their behaviors. In order to further support this, like I was going to do, we need to use in situ hybridization and or acridine orage to physically see the evidence of the damage compared to the other time points. And a cortisol analysis of the adult fish would also be critical to further support that they're not actually feeling stress and there's not another thing happening. So this all would have been completed if it weren't for COVID-19 outbreak. It's disappointing, but this definitely leads the way for other people to conduct further research based off this paper. And the phototaxis behavioral assay did not show significant results, as I stated previously. In contrast with the research conducted by Bai et al. 2016, the negative phototaxis was observed, as I stated earlier, instead of positive. And a couple reasons could be for this. They didn't use an LED light like I did in their study. They used a normal incandescent. So that could be a factor. It could have been a little bit brighter than theirs. And it also could be they did use slightly older zebrafish around. They used 2 to 3 week old, which I used only 6 to 8 to 10 day old zebrafish for my studies. So it could just be a developmental delay. So if I were to go back and do this again, I would put them at that time point to see if it's actually just it takes a while to develop that phototaxis result as opposed to the thigmotaxis result. All three thigmotaxis groups showcase the devastating effects of alcohol exposure. So it really does show that, because all the groups, regardless, for the thigmotaxis test, the treated groups were so much lower than the control groups in their stress response, which means there is damage. So it really does show the devastating effects of alcohol exposure to a developing organism, and it applies to humans as well, not just zebrafish. With these results, I found further knowledge can be given to mothers on the dangers of drinking during pregnancy, and it's really we could do further research on what specifically alcohol targets and how we could potentially fix it, with the different treatments we could create in the future to target the HPA axis. And these are all my sources. There's a lot of them and they're hard to read. And I'd like to thank you all for attending this and listening to it. I know this all has been crazy. So thank you so much for listening to my presentation.